There you go. Hello everyone, this is Mr. Olson.
And I'm Mr. Sherping, Mrs. Nestinen, student teacher.
Perfect. So today guys, before we even start lab, I want you guys to pause this video and make this chart in your notebook. Okay? It says sample, protein, starch, sugar, positive, negative, and then 1, 2, 3, and 4. So put this in your notebook. I'm going to pause the video, or I'm going to stop it, and then we're going to set up some lab. That's what we're going to do today. Sound good? Awesome. Is it going?
Yep.
Perfect. So today, guys, the first thing we're going to test for is going to be proteins. So we're going to run a couple negatives and positives first just to kind of test to make sure our experiment's going to work properly. So I have distilled water, which is going to be our negative control because it should have no proteins in it. Then I'm going to have our positive control, which is our protein, which is just realistically whey protein powder. So we should know this has protein powder in it. Now, we do have two beakers here. One says negative, one says positive. I'm going to add about a dropper full of my distilled water into my negative beaker. I'm going to add about a dropper full of my positive protein into my positive beaker. And now, guys, realistically, the reason we're doing this is just to tell if our experiment's actually going to work by doing the process that we're trying. So if it doesn't work, that means we have an issue in our system, which means we need to probably change it. Okay. So I'm going to start off with my positive, and for this, we're going to add one dropper full of sodium carbonate, which is this clear solution. So I'm going to add this one whole dropper full, dump it in. And then I'm also going to add 2 to 3 drops of this copper sulfate, which is this light blue solution. Take some of this. I'm going to add my 2 to 3 drops. 1, 2. Shake it up. And as you guys can see, it turned purple. A purple means it is a positive test. Now, we're going to refer back to this, guys, for the scale when we do our unknowns. Now, as for the negative, I'm going to do the same exact thing. Take a whole pipette full of sodium carbonate, put it in there, add a couple of drops of this solution, shake it up. As you can tell, guys, it stays clear. Okay? Now, this is the positive. This is for sure protein. This is our negative, proving that there's no protein in it. Okay? Now, we're going to do this for our 4 unknowns. Now, give me 1 second. I'm going to set those up. We're going to keep going. Perfect. We're back. So, guys, we have put together unknown 1, which is this yellow solution. Unknown 2, which is this solution. Unknown 3. And unknown 4. So we're going to test these solutions to see if they have protein in them. That way, once we do protein, starches, and sugars, you guys should be able to make a guess on what we're actually looking at. So, unknown 1 is right here. It's slightly yellow. I'm going to do the same thing we did with our controls. So I'm going to add a whole pipette full of sodium carbonate. Then I'm going to do a couple of drops of my copper sulfate. 1, 2. I'm going to shake it up. And nothing really happens, okay? And we're going to come back to that. So let me turn my water down, guys. Now I'm going to do the same thing with unknown solution number 2. Whole pipette full once again. A couple of drops of copper sulfate. Shake it up. And it started changing color. Okay, so we have some type of positive testing for proteins. Do this for number 3. Pipette full of copper sulfate. This isn't copper sulfate. This is sodium carbonate. I'm sorry. Couple of drops carbon sulfate, blue solution. Shake it up. And actually get partially a positive test. Okay, turns out to be blue. And now we're going to do our 4th one. Okay, once again, sodium carbonate, whole pipette full, copper sulfate, a couple of drops. Shake it up. Once again, we have a positive test. A little bit lower. Is that good?
Yeah.
Perfect. Okay, sorry if you guys couldn't see that. But now we have another positive test. So we have a variety of different test results here. Okay, varying anywhere from 1, which is this, 2, which is this, 3, which is this, and then we have 4. Now, guys, in this meantime, I want you to take out your notebooks and go to that chart, okay? We're going to look at that chart together.
All right, so now that we're at our chart, we're going to take a second and fill it out based on the samples that we have. So first, we're going to start with our positive and negative test. So we know that our positive test has protein powder in it. So we know there's a bunch of protein in here. So we're going to kind of rate each of these samples by how much protein they have based on the color that they turn. So since we know we have a lot of protein in here, this is basically just whey protein powder. We're going to give this a score of 4. So our rankings today, we're going to start from 0 to 4. 0 means that there is no indication, there's no color change, meaning that there's no protein. And 4 is going to be our maximum change, showing that there's a bunch in there. So since we know that there's a lot of protein in our positive test, we're going to make it 4. And then our negative, you can see there wasn't really much of a change. There's a little bit of blue because our indicator was blue, but you didn't really see a drastic color change. So we're going to call that 0. So now that we kind of have some reference between how each of them look like, here we have unknown sample number 1. We don't know what this is. You can see that it didn't really change color at all. It's still pretty clear, meaning that we didn't really have any protein in it. So I think we call this one 1 or 0. So I'm just going to give it a 1. There's a little bit, but not too much. Unknown sample number 2. We can see that there's a pretty big drastic change. It turned pretty yellow, or not yellow, sorry, purple, but not as much as our positive here. So a positive is super purple. This one's a little more light. So for number 2, I'm going to give it a score of 2. These scorings are just subjective, guys. It's not like we need to have a 0, a 1, a 2, or 3, or 4. We're just kind of giving it our best judgment. So number 3 here, we can see it's more purple than our number 1 here, but it's not quite as purple as number 2. So I'm going to give it a score of around 1, since it's more on the blue side. And then finally, our number 4 turned really purple here. It's still kind of clearish. It's not like the super thick purple. So I'm going to give it a score of 3, just behind our positive test of the protein powder. So that's basically how we're going to rate all of our sample tests here for the rest of the experiment here. So right now we're going to go on pause, and we're going to fill up our test tubes with the other solutions and start testing for our starch.
Welcome back, guys. We're going to test for starches. So once again, we have our negative control, which is just distilled water. And this time, we actually have our positive control, which is just starch that you would use for like your clothes, which I'm sure none of you guys know what that means. But back in the day, you used to like starch your jeans. Sure, I guess. But we're going to test it. So to test this, guys, we're going to use Lagul solution, which in realistic terms is just iodine. That'll tell us if something is has starch or has no starch. Now, this is a brown solution, so it's going to turn some things partially brown, so take that into note. But first, we're doing negative solution. We're going to add a couple drops here. I'm going to shake it up. All it did is just turn that clear water brown. This is negative. This is just proving that this is a negative test. Okay. Now, this, on the other hand, should test positive. So let's see what this does. So once again, we're going to add 1 to 2 drops here. 1, 2. Shake it up. As you guys can see, it's a pretty drastic change. Okay. This was pretty much white, and now it's a really dark purple to almost black. Okay. So this is going to be like our 4-star rating when it comes to starch solution. This is a high starch solution. Now, we're going to do the same thing once again with our unknowns. So once again, guys, we have 1, 2, 3, and 4. Same unknowns from last time. Okay, so these are the same ones that go in your chart. So unknown 1, I'm going to grab some of this Lagul solution. Pull this over here. Pull it over there. Pull it over there. Grab this. Add 2 drops. Boom, boom. Shake it up. And once again, this realistically didn't change at all. Okay, and you guys will get a better image of this as well. Okay, but it's still just normally brown. We're gonna do unknown 2. Add 2 drops. 1, 2. Shake it up. And bam, big change. Okay, so once again, this big change here is testing positive for starch. Okay. Now let's do this for number 3. 1, 2. And this had a huge change for starch as well. So unknown 3 has a lot of starch. And let's do the last one, number 4. 1, 2. Shake it up. And this one doesn't change at all. Okay, so this is a negative test for the unknown. Now we're gonna set this up, guys, so you guys can see the whole variety in color with the positives and negatives. Give us 1 sec. Welcome back, guys. Trying to give you the best image of what these solutions are showing. So Mr. Sherping's going to show you the positive and negative just to remind you. So there we go. The purple, that dark purple is the positive. That yellow is the negative test. So looking at our unknowns, we can look at unknown 1, unknown 2, unknown 3, and unknown 4. Unknown 1 is this yellow. There we go. Unknown 2 is this purple. Unknown 3 is this purple. And unknown 4 is this clear solution. What you guys need to do, pause the video, fill out your chart, whether it be a 1, 2, 3, 4, or a 0, and put that in the same chart we just did for our proteins. Do it for the starches. Ready? Go. Welcome back, guys. So in this one, we're testing our last thing. We're going to test sugar. So we're going to use Benedict solution, and we're also going to use a hot water bath. Okay. So I've already ran the experiment. I've already done the same exact things. We have our positive. We also have our negative control. So our negative control, once again, is distilled water, which shouldn't have any sugar. Our positive control is just some sugar in water. Okay. So let me grab the positive and negative controls quick. So there's that. Where's the negative? There it is. So my positive control turns orange. Okay. This proves that we had a color change. This changes. Guys, it's the same yellow substance with just blue added to it. This turns bright orange. This is a 4-star rating for sugar. Now, our negative, our distilled water is just blue. Okay, so it's blue with just clear in it. This proves that there's no sugar inside of our water. Now, let's look at unknown substance number 1. So unknown substance number 1 looks pretty similar to our control. Looks pretty similar to our control positive. So this does have a high concentration of sugar. Number 2 is also pretty high concentration of sugar. It's pretty orange still. Number 3 still has a color change, but not as drastic. It's not as orange. It's more of a green kind of vomit color. And then number 4 here, turn almost like a purplish dark blue. Okay, this is pretty close to a negative test for sugars, but it is showing that there's probably still some in there. Okay, so give us 1 second, guys. We're going to show you the huge variety of them again, and this will be the last thing we do for today. Perfect. So here, guys, we have test tube 1, 2, 3, and 4. 1 is over here. Okay, by this hand that's wiggling. Test tube 1, test tube 2, test tube 3 and 4 over here. What I want you guys to do is fill out your chart with your 4s, 3s, 2s, and 1s, determining which one has the most sugar, which has the least sugar, compared to our positive and our negative control. Other than that, guys, this is really all we're going to do today. This is what lab is. It's really just testing proteins, carbohydrates, and starches. Guys, make sure you fill out your post-lab questions. If you guys have any emails, please reach out to us. Email your recording teachers. Other than that, guys, have a good day. Bye.